Okay, so hi everyone. My name is Nellie, and I'm a graduate student in the labs of Vivek Swaroop and Kim Green. And today I'm going to be giving a brief introduction to spatial transcriptomics. So this is like a very entry-level, high-level introduction to spatial transcriptomics. I'm by no means an expert, but yeah, hopefully this is helpful. Here are all the things I'm going to touch on today. So I'm going to introduce transcriptomics in general, then I'll talk about some available technologies in the field, and how you decide which technology to use. I'll talk a little bit about analysis, and then just kind of summarize at the end. And then after that, I also wanted to share some data that we have from Model AD that I've been analyzing over the last few months. And yeah.
Stop recording.
Yeah. Yeah.
Let me know when.
Okay, cool. So first I'll just talk a little bit about what what transcriptomics is. So in order to talk about transcriptomics, I need to first define what omics is. So on the left, here's a picture of the central dogma, which everyone knows. So there's DNA that's transcribed into RNA, which is translated into protein. So omics is the comprehensive study of molecules within biological systems. And so genomics is the study of DNA or genes. Transcriptomics is the study of RNA transcripts. Proteomics is the study of proteins. Metabolomics is the study of all the metabolites. And epigenomics is the study of epigenetic changes, including DNA methylation and histone modification. Oh my God. So today I'm going to be mainly talking about transcriptomics. In this field, there's a lot of focus on transcriptomics because genomics, which is just the study of DNA, will not tell you a whole lot by itself because like the genome is same from cell to cell, because all cells have the same DNA. So it's really the different levels of RNA transcripts that are different from cell to cell. And really our end goal is to look at proteomics, which is all of the proteins in the cell, but that's a little bit harder for us to quantify, at least with the current technology that we have. So transcriptomics is kind of a good intermediate. So now I'll talk a little bit about a timeline of transcriptomics technologies over the last few decades. So in the 1990s was kind of the first, was when we first started having microarrays. So microarrays basically give you a broad view of gene expression across many genes simultaneously. So if you imagine like an array with a bunch of spots, each spot will have like a different probe for a different gene of interest. And then you can like just put your sample on this microarray and then see what different genes are being expressed. So that's microarray. And then in the 2000s, they came out with what is now known as bulk RNA sequencing. So this basically involves sequencing actual RNA molecules, whereas with microarray, you have like probes hybridizing to your genes of interest. So bulk RNA is a lot more detailed and quantitative, but bulk RNA sequencing takes like your whole sample together. So you're not looking at single cell resolution at this point. So you're still missing a lot of important information if your sample is heterogeneous. So in the 2010s, we started seeing more and more single cell RNA sequencing. So this really allowed us to refine the resolution to single cell. So every single cell is considered, like its transcriptome is considered on its own. And so we've seen a lot of advances in science because of the advent of single cell RNA sequencing over the last decade or so. And most recently, in the last several years, has seen the rise of spatial transcriptomics. So not only do you get single cell resolution or close to single cell resolution, but you also get this added dimension of spatial context. So you can see like where cells are in position to one another, in relation to one another. And then you can also look at neighborhoods of cells adjacent to other neighborhoods and see the differences in their transcriptomes. For those of you who are not super familiar with transcriptomics technologies, I've kind of like adapted this analogy from a talk that I attended. But I kind of added like this fruit platter thing. So imagine you have a platter of fruit, and on this platter of fruit, you have like, it's like very nicely arranged, and you have like watermelon on one side and oranges on another side and like grapes in the middle and like strawberries throughout. So first imagine you take this fruit platter and you put all the fruit into a blender, you blend it up and you make a smoothie. So that's called bulk RNA sequencing, where you blend all of your fruit together. So you lose the individual identities of the fruits as well as their spatial arrangement on the original fruit platter. Next, imagine you take the same fruit platter and you dump all of the fruit from the platter into a bowl and you mix it up. That's called single cell RNA sequencing. So now you're able to taste each individual fruit separately, but you still lose the original arrangement of the fruit on the fruit platter. And then finally, obviously the last one is spatial transcriptomics. So now you're appreciating the fruit platter as a whole. So you can taste each individual fruit, and then you can also appreciate the arrangement of the fruit in space. So in this analogy, every piece of fruit is a cell, and then the fruit platter is your tissue sample, whether that's brain or liver or lung or whatever you're interested in looking at. Also, I made these images with ChatGPT, so sorry if they're a little creepy. Okay, so now I'll talk about different technologies available for spatial transcriptomics. So broadly, there are two classes of spatial transcriptomics. There are imaging-based technologies and sequencing-based technologies. These are also called capture-based technologies. So imaging-based technologies are targeted or biased, meaning you have pre-selected targets. These can either be selected by you, the researcher, or they are like curated by whatever company you're working with for whatever platform you're using. So you can have like a panel of genes specific for neuroscience or immunology or oncology or whatever your specific experiment is. The benefit of using imaging-based technologies is that you can get very, very high resolution because it's imaging-based. So you can see within each cell where transcripts are being expressed. By contrast, sequencing-based technologies are what's called untargeted or unbiased. So you can actually get the entire transcriptome. So this is much higher throughput. However, sequencing-based technologies will usually lack single-cell resolution. So you'll have like a specific spot size, which may contain like 10 to 50 cells within that spot. So there's trade-offs to both of these technologies, and we're not really at a point yet where we kind of have the best of both worlds with like super high resolution and super high throughput and high sensitivity. So here's an example of imaging-based transcriptomics. So this is MERFISH from VizGen. So basically, it's because it's an imaging-based approach. You can see like individual transcripts being expressed in this cell. So each colored dot is like a different RNA transcript. But you can see here that you can only profile 483 genes out of the whole genome. So that's only a small fraction of the whole genome. So this is subcellular resolution, but it's targeted or biased. And so this is Visium, which is a sequencing-based technology. So here you can see that you're sequencing the whole transcriptome, but you're not at single cell resolution. So your unit is basically like one spot. So you'll have multiple cells within that spot. So this is whole transcriptome or unbiased, but it's lacking single cell resolution. So just to kind of further break down these two methods, there are imaging methods and sequencing methods. Within the imaging-based, you have in situ hybridization-based and in situ sequencing-based. So with in situ hybridization-based, you basically have these probes that you design. And then the probes will have fluorophores, and then they'll bind to your tissue. And then for every probe that binds, you will like take a picture of your tissue, and then you'll repeat this however many times it takes to get a full barcode of that RNA transcript. And yeah, so that's basically how hybridization-based imaging methods work. So this is used in things like COSMX, which is by nanostring. There's also in situ sequencing methods. So this involves same thing, you design a probe, and then you bind the probe to your target tissue, and then you do what's called rolling circle amplification to basically amplify your transcripts. And then you will sequence within that rolling circle. And then as you sequence that rolling circle, then you will image at each like transcript, and then same thing, you'll repeat as many times as necessary to get the full sequence. And then for sequencing-based methods, there's also two kind of subcategories. There are array-based methods and microdissection-based methods. So for array-based, you basically have an array of barcoded probes, and you will like overlay your tissue on top of this array, and each spot has its own unique barcode. So when you lay your tissue on top of that array, then each spot on your tissue will have a spatial barcode. And then you will sequence each spot with next generation sequencing. But the downside of this array-based approach is that you're not getting single cell resolution. And there have been attempts to like reduce the size of the spots in the array. But even if you were to reduce the size of the array to like single cell size, it's not actually a single cell. Like you might have one cell that's overlapping two spots. So even if you were able to reduce the size significantly, it still wouldn't be true single cell resolution. And then the final approach is called microdissection. And so basically you hybridize your probes to your tissue, and then you choose a spot that you're interested in, and you release the probes from that spot using UV or other approaches. And then you capture all of the RNA transcripts in that specific spot, and then you sequence that spot, and then you choose another spot and you sequence that spot. So those are the four like main types of spatial transcriptomics technologies available right now. So here are the current technologies in the field. This is not all of them, but these are kind of the commercially available ones. So you might see some familiar names. We have all of these on campus, as far as I know. And so they each have like different pros and cons. For example, Visium and Xenia. Oh, so Visium is sequencing based. Geomics is also sequencing based. But the specific technologies are slightly different. So Visium, I think I showed a picture of this earlier, but like it's array based. So you'll have a spot, and the spot is 55 microns in diameter. But this approach allows you to sequence the whole transcriptome. And then geomics is slightly different. It takes a microdissection approach. So it'll take each spot and sequence that spot separately. And then you have Xenium, which is in C2 sequencing based. So that's what I talked about earlier with the rolling circle amplification. And then there's Merscope and Cosmix, which both kind of have similar technologies, but these are imaging based, and they use in situ hybridization. So basically you have a specific probe that binds to the tissue, and then it'll like release fluorophores at each like step, and then you take pictures of that fluorophore. And for all of these platforms, most of them can take basically any sample that has viable RNA in it. So it's pretty flexible, which is nice. And then some are whole transcriptome, which is WT, and then the others, like the numbers, I've listed there if you're interested. Okay, so now I'll talk about how to design a spatial transcriptomics experiment and things to consider. So one of the main questions you should be thinking about at the beginning of your experiment is what your experimental aim is. So if you are taking more of like a discovery approach, where you want to generate a hypothesis and you don't really have a question at the beginning, it's better to use a sequencing-based approach, which is unbiased and untargeted. So you have like all of the transcripts in your cell, and you may not have like single cell resolution, but at least you're getting all genes, and then you can kind of go from there. Whereas if you have a specific question in mind and you want to test a particular hypothesis, if you have like specific gene targets you already are interested in, then in that case, an imaging-based approach might be more appropriate, where you have a select panel of genes, but you're able to see at very high resolution. Some other technical considerations are sample number. So each of these different platforms have different like sizes of like imaging area or sequencing area. So you want to take that into consideration as well. And then mRNA capture efficiency. So some, like for example, sequencing-based technologies tend to have lower capture efficiency than imaging-based. So imaging-based can capture like almost all of the transcripts that are being expressed, whereas sequencing-based technologies don't have that capture efficiency, and you might actually miss a lot of like lowly expressed genes. Another thing to consider is spatial resolution. So do you care about seeing individual cells and where transcripts are being expressed within each cell? Or would you rather just see like broad, like spatial neighborhoods? And there are also ways to increase the resolution if you choose like a sequencing-based technology. And then the last consideration is number of genes profiled. So are there specific genes you are interested in looking at, or do you want to just profile everything? So next, I'll talk about tools that are available for spatial transcriptomics. So for those who are unfamiliar, I've included the single-cell RNA-seq pipeline here. The pipeline is pretty similar, but basically at the beginning, you get your sequences and you want to align them to a reference genome. And then you're going to make this gene by cell or cell by gene matrix and quantify your transcripts that are being expressed. Then you want to do quality control and filtering. So you want to filter out anything that is like empty or doublets or just like dying cells. Then you'll normalize all your data, do feature selection, reduce the dimensions. So every single gene and expression of gene is like one dimension. So you have like thousands of dimensions. So you want to reduce that down to something that we can easily understand. Then you want to do visualization and clustering. And then from there, after you've done like that kind of standard pipeline, then you can do a lot of cool downstream analysis. So the pipeline for single-cell RNA-seq is pretty similar to that for spatial transcriptomics. One big difference is at the beginning in the pre-processing stage, but thankfully this is typically built into your platform, whatever platform you're using. So for imaging-based spatial transcriptomics, you will have a ton of images, like terabytes of images, and you need to align those images at each field of view and filter them. And then an important step of imaging-based spatial transcriptomics is called cell segmentation. So like for a particular field of view, you'll have a bunch of transcripts that are being expressed in space, but without doing cell segmentation, you have no idea like what is within one cell. So they have developed these algorithms to basically segment cells. So this is kind of like the key feature of imaging-based spatial transcriptomics is being able to segment individual cells. And this is kind of a big challenge. Like it's difficult to do this. Yeah. And then for sequencing-based technologies, you want to align your sequences and then map those sequences back onto space. So that's the pre-processing stage. Thankfully, we don't usually have to worry about this. Like this is all done by the computer. And then in the processing stage, these steps are pretty similar to the previous slide, but we'll do QC and filtering, normalization, identifying variable features, scaling, dimensional reduction, and clustering. And this can either be done by like your whatever platform you're using's like proprietary software, or you can just export this pre-processed data and do these steps yourself. And then downstream of that, there are a lot of cool analyses you can do. One important thing to do for array-based technologies is something called deconvolution. So with array-based technologies, like within a given spot, you will have multiple cell types. So if you're looking at the brain, you could have like neurons and oligodendrocytes within the same spot. So deconvolution is basically a way to account for that. I've never done it, so I don't actually know like the details, but that's basically an important step for array-based technologies. And then obviously you want to identify what your cell types are. You can also, with spatial data, identify different spatial features and spatial neighborhoods. And you can do differential expression analysis, similar as single-cell RNA sequencing. You can also do differential localization analysis. So like, how does one spatial region differ in its transcriptome from another spatial region? You can also do cell-cell interaction. And then finally, you can integrate all of this spatial transcriptomics data with other omics data. So you could integrate this with like single cell RNA sequencing data or ATAC-seq data or proteomics data. So here are some toolkits that are currently available. The first two you've probably heard of if you have ever done single cell RNA sequencing analysis. So these are Seurat and ScanPy. Yeah, they were originally made for single cell RNA sequencing, but they've recently added functionality for spatial transcriptomics. And the benefits of these two toolkits is that they have extensive documentation, so they're pretty beginner friendly, I would say. So I use Seurat, and I consider myself a beginner, but I feel like it's been pretty straightforward for me to like analyze all of this spatial data, just using their standard pipeline. And then below, there are a lot of other toolkits you can use. And these are specifically made for spatial transcriptomics. And I've listed here the different languages and platforms that are compatible with these toolkits. And yeah. So these tools and many more are still in active development. So when I first started analyzing the spatial transcriptomics data, I don't think Seurat had any like spatial tutorials. So I was basically just treating my data like a single cell data set and then just like adding in some spatial stuff. But in the last few months that I've been analyzing this data, they've come out with new tutorials specifically for spatial data, which is pretty cool. Yeah, so the future of spatial transcriptomics. So all of this technology and all of these pipelines are very rapidly evolving. And in the near future, I think the goal of all of this research is to be able to combine single cell resolution with whole genome scale profiling at high sensitivity. So we want to be able to see individual cells. We want to profile all of the transcripts that are being expressed by a cell. And we also want to see all of the transcripts, not just the highly expressed ones. So one challenge of spatial transcriptomics in general is that we are producing a ton of data with every single experiment. So I think one bottleneck will be like on the computational side, like how do we analyze all of this data?